To another A-level biology revision video of Mr. Phillips, and today we are going to be looking at the nitrogen cycle, which forms part of the energy transfers and nutrient cycles module. So if we get into it, you need to be aware that what we're talking about are natural cycles. We will mention human additions to them, but we're talking about things that are not being changed by humans overall. And we're going to look at nitrogen, then in the next video will be the phosphorus cycle. They naturally occur in the food chain. Human activity can disrupt them, but they're not part of what we're studying. So before we introduce the nitrogen cycle, we need to look at the role of microbes in cycles. So there's two types of microbes you really need to understand. The first one are saprobionts, and saprobionts can be either bacteria or fungus, okay? And basically a saprobiont is something that feeds on the remains of the dead animals and organisms and on their waste products. So effectively the decomposers that we are absolutely vital to have in our ecosystems, and they get rid of waste products, break it down into inorganic molecules. And they use something called extracellular digestion, which is where you release enzymes to the external environment. The enzymes, let's say protease, then digest the protein into amino acids, and then they can absorb simpler molecules as a result. And that's called saprobiotic nutrition. And the other important microbe that we're going to introduce might be new to you today is something called Mycorrhizae. A mycorrhiza is a fungus that forms a symbiotic relationship, or sometimes known as a mutualistic relationship, with roots of the plants through fungal hyphae. So if you know anything about fungus, they've got like tiny, almost root-like structures themselves, but obviously a lot smaller, which sort of detect what's going on, like they feel for the environment and absorb stuff through. And the fungal hyphae form a link with the roots of the plant. Now this is good for the plants because it massively increases the surface area of the fungus, help increase the surface area, which increases the mineral ion uptake, particularly of really like low level minerals in the soil like phosphorus. It also helps the absorption of water. For the fungus, the advantage is that the plant will give it some organic molecules such as glucose as a result. So both of them get an advantage, and you can see the picture there that you've got a plant with mycorrhizae. A lot more, looks like it's got a lot more root in there, but it actually hasn't. That's a lot of the fungus and the hyphae of the fungus. So the nitrogen cycle, then going on to the nitrogen cycle is really important, and the reason the nitrogen cycle is important is obviously if you hopefully think all amino acids contain nitrogen, but the nitrogen is an issue because whilst it is absolutely vital for life, and obviously makes proteins, so amino acids, proteins, also nitrogen in DNA. 78% of the gas in the air is nitrogen, but it's really hard to actually break it down. So we breathe in 78%, we breathe out 78% nitrogen, and that's because nitrogen's got a triple covalent bond, which is very, very strong, and it takes a huge amount of energy to break that. So most organisms can't absorb it and use it from the air, and that includes most plants and animals. So very few organisms can actually access it. And what has to happen is it needs to be reacted in or converted into a more reactive form of ammonia or nitrate in something called nitrogen fixation, and we're about to look at very few bacteria are able to do. So then here we have the nitrogen cycle. Now you need to learn this off by heart and then be able to add the extra detail of what extra things are sort of happening as we go through. So what we're going to look at is we're going to start off by looking at nitrogen, what we call nitrogen fixation. So if we start with the nitrogen in the atmosphere, okay, it starts in the atmosphere, so at the top of this diagram, which you need to really learn in detail, you've got nitrogen there, okay? Now nitrogen then can come out of the soil and, out of the air and into the soil using a couple of methods. The main method of which nitrogen is going to come into the soil is through nitrogen fixation using something called rhizobium. So down this left-hand side here, and that's where 90% of the nitrogen enters our soil. And what rhizobium are is a special type of bacteria that have the ability to fix nitrogen from the air. So it's carried out, as I said, by this bacteria called Rhizobium, and this turns nitrogen into ammonia, and then that goes on to form ammonium ions in solution that can be absorbed by the plant. Now, the Rhizobium are bacteria that have, again, a mutualistic relationship, and they're found in the root nodules, which are like parts, little growths on the root of particular plants. And what we call these plants are called leguminous plants, and it's things like beans, peas, clover, so things that are very, very healthy for us. And again, just like before, the plant's going to provide the bacteria with glucose, whereas the bacteria is providing the plant with that essential nitrates, which allow the plant to grow. There are other nitrogen fixating bacteria also found in the soil, and therefore also nitrogen fixation can happen by other bacteria in the soil. And there is extra processes as well, so 90% of it happens by Rhizobium bacteria. We do it ourselves in the Haber process. It's obviously an artificial process, so again we convert nitrogen from the air, we mix it with hydrogen using the Haber process in very specific conditions, if you remember your GCSE, and that then turns that into ammonia, which can be used on the soil as fertilizer. We can also use, we don't also use another natural process, is nitrogen fixation by lightning. The amount of energy in lightning can split nitrogen into two, and then that turns into nitrates as it reacts with the oxygen in the air to form the nitrates there. Okay, so we've got it coming out of the air and into the soil using nitrogen fixation by either Rhizobium bacteria, other bacteria in the soil, the Haber process, which we're not going to talk about because it's not natural, and lightning. So what happens next then? Well, if we start our journey in the plant, which has obviously now got nitrogen from the bacteria, so it's in the plant, okay, and the plants then are eaten by animals, okay, and the plant is eaten by animals, and the plant or the animals can die, okay, so they can die and go down this route here, or they can just excrete urine into, or urea into the soil in urine. Okay, now what then happens is a process called ammonification. Okay, and ammonification is basically where in the soil there we have the organic nitrogen that comes into the soil, which is so the dead bits of animal and plant, or dead bits of animal and plant, and then you can have urea, which also comes obviously from our urine, and that can be turned into ammonium, okay, and that is done by saprobionts, what we talked about earlier on, about the saprobionts, the decomposers breaking down the organic compounds into simpler inorganic ammonium. Now, once that happens, we can then go down a series of reactions, breaking the ammonia down into nitrites and then nitrates, and that is oxidation reaction. If you look at that, you're adding oxygen to into nitrite and adding more oxygen in to turn it into nitrates. Now, nitrification is basically done in two stages. So there's something called Nitrosomonas bacteria, which do the first part, and then Nitrobacter that do the next part. So that is important that you learn those terms. And the nitrates then can do one of a couple of things. So the nitrates can be reabsorbed by plants. So I'm just going to change my colour here to hopefully make it sort of obvious to you. So nitrates can be reabsorbed by regular plants and also obviously leguminous plants, and that can go back into the plants, into the systems. The nitrates can leach off and go into rivers and streams, and that can obviously cause issues if it's too much. If it's from fertilizers, that can lead to eutrophication, which we'll talk about in a few less time, or it can just happen naturally. It's not a massive issue. The other thing that can happen though is denitrification. So some of those nitrates can go back into the atmosphere. Okay, and the atmosphere, obviously, if we're doing denitrification, we're taking nitrogen out of the soil. Now, denitrification is when the nitrates then are converted into nitrogen gas. Okay, and they use nitrates in the soil to carry, and it's done by bacteria, denitrifying bacteria, which are basically doing anaerobic respiration using nitrogen, or the nitrates as the respiratory product. the respiratory substrate. So therefore, respiration is carried out, it produces nitrogen gas, and that is released into the atmosphere. It happens only in anaerobic conditions. So something like waterlogged soil would be somewhere where this bacteria is able to do this. So that is the overall nitrogen cycle, and what you need to be able to do is explain all those stages. So you've got to be able to explain nitrogen fixation, ammonification, nitrification and denitrification. You need to be able to name the types of bacteria that are on this picture. So really essential to learn it and be able to talk your way through. So just to add a little bit more detail to a couple of points then, so we said first of all nitrogen fixation is where we turn nitrogen from the air into ammonium and then into nitrates. So that was done by Rhizobium, which are bacteria that lives in the legumes such as peas, and you can see here on this micrograph that you've got you can see the bacteria on the roots of the organisms, and here you can see the on the legumes that there is the nodules that the bacteria can live in. And what happens, as we said before, is that the bacteria, when the bacteria germinates, the Rhizobium attaches itself to the seed, and then it will very quickly get into the roots. They can't be very successful in free soil, but they're much more common in leguminous plants like peas. Okay, so how does the Rhizobium do it? Maybe a bit more than you need, but effectively what the Rhizobium does is it takes the nitrogen, and think about what ammonia is. Nitrogen is NH3, so you have to be adding hydrogen to that. So the Rhizobium use energy in the form of ATP, and as they do that, they take a reduced coenzyme and they obviously add hydrogen to that. So the hydrogen goes in to make NH3, oxidizing the enzyme that can be then reused again. But this will only happen in anaerobic conditions, and it relies on neutralization. Fixation can also occur, as we said, with lightning, okay, just because the amount of energy can you use to split the nitrogen up, and then it reacts with oxygen to make the nitrates, dissolves in the rain, and that happens quite a lot in tropical countries while there's a particularly sort of damp, particular smell when there's thunderstorms. The harbour process is obviously also a way, but it's not a natural way, so we're going to focus on that. So then returning to the soil, so once the plant or the animal dies or it produces urea, this can be broken down into something called ammonification, which is when the saprobionts break down the proteins back into ammonia and into nitrates. So first they digest proteins and amino acids, and then they remove the amino groups from the amino acids using enzymes to do so. Then we've got nitrification, and nitrification, if you remember, is where we take the ammonium ions and we turn them into nitrates, and then the plants can reabsorb them from the soil. So what we do is we take the ammonia, we add oxygen to it, we oxidize it to make nitrite, and then we oxidize it again to make nitrate. And if you remember, there's two types of bacteria that do that. The first type of nitrifying bacteria, which would turn it from ammonia into nitrite, is Nitrosomas, and then nitrobacteria, or nitrobacter, turn it into nitrate. Okay, now the bacteria use this to do chemosynthesis, basically their version of chemical respiration, and obviously plants can only take up, they need to have that more reactive form, so they can only take up nitrogen in the form of nitrates. So this is essential for plants and the recycling of minerals. And this bit can only happen in aerated soil, which hopefully makes sense because you need the oxygen in aerated soil to make ammonium to become nitrite to become nitrate, because you're adding oxygen. Now the last stage is denitrification, and we said this happens in anaerobic conditions such as waterlogged conditions, where denitrifying bacteria convert nitrate into nitrogen, which is then lost into the atmosphere. And this is really useful in sewage treatment and compost, because it helps break down sewage, and it also allows us to work with it in anaerobic conditions. So again, really, really key, you learn the 4 different stages. So think about it in terms of 4 stages. If we start off with The first stage, which is nitrogen fixation. Okay, so nitrogen fixation is these ideas here of the ribosome and bacteria, and these processes here of hard process and funda. You've then got ammonification, which is where you take that urea and ammonia, you take the proteins in the plants and animals, and you take that and turn it into ammonia. You then have denitrification, which is where you take the ammonia and convert it into nitrites using Nitrosomas, and then nitrate using oxidation, due to Nitrobacter. And the last one is denitrification, I've got the wrong way around, and that one is where you take the nitrogen out of the ground back in using anaerobic bacteria. So really essential that you absolutely learn the stages of this cycle, what bacteria do what, and just learn the different stages of it, so in terms of being oxidized or being reduced, to ensure that you can fully follow the cycle. So it's been useful, and I'll see you next time for the phosphorus cycle.